Restriction enzymes are proteins found in bacteria that cut DNA at specific target sequences, resulting in the formation of smaller DNA fragments. These enzymes are commonly used in the field of biotechnology for various applications, such as DNA cloning, gene editing, and genetic engineering.
In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in Escherichia coli were isolated. One of these added methyl groups to DNA, while the other cut DNA. The latter was called restriction endonuclease.
The first restriction endonuclease HINE2, whose functioning depended on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that HINE2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs.
This specific base sequence is known as the recognition sequence for HINE2. Besides HINE2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognizes different recognition sequences.
The convention for naming these enzymes is the first letter of the name comes from the genus and the second 2 letters come from the species of the prokaryotic cell from which they were isolated, e.g. EcoRI comes from Escherichia coli RY13. In EcoRI, the letter R is derived from the name of the strain. Roman numbers following the names indicate the order in which the enzymes were isolated from that strain of bacteria.
Restriction enzymes belong to a larger class of enzymes called nucleases. These are of 2 kinds, exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make cuts at specific positions within the DNA.
Each restriction endonuclease functions by inspecting the length of a DNA sequence. Once it finds its specific recognition sequence, it will bind to the DNA and cut each of the 2 strands of the double helix at specific points in their sugar phosphate backbones. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence in the DNA.
Do you know what palindromes are? These are groups of letters that form the same words when read both forward and backward, e.g., Malayalam. As against a word palindrome where the same word is read in both directions, the palindrome in DNA is a sequence of base pairs that read the same on the 2 strands when the orientation of reading is kept the same. For example, the following sequences read the same on the 2 strands in 5'3" direction. This is also true of Fred in the 3'5" direction.
Restriction enzymes cut the strand of DNA a little away from the center off the palindrome sites but between the same 2 bases on the opposite strands. This leaves single-stranded portions at the ends. There are overhanging stretches called sticky ends on each strand. These are named so because they form hydrogen bonds with their complementary cut counterparts. This stickiness of the ends facilitates that action of the enzyme DNA ligase.
Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources/genomes. When cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends and these can be joined together, end-to-end, using DNA ligases.
You may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created.